I want to talk today about our patient-derived xenograft models and how we enhance such program and such new models. So just as a first introduction, as you all know that cancer pathology remains a major cause of death worldwide, and there is a high pressure on the healthcare system, but also in the developmental costs to push for new compounds in clinical. So now we have new treatments, but the former treatment remain inefficient, and new development are focused on targeted therapies. The main standard now is to go towards personalized medicine, and for this we need highly characterized models, and that represents the high variability of tumors. And the standard cell line that we have used for a while now might not be very representative of the pathology and the diversity of the pathology, cancer pathology. And there is a new need also is to associate all this pathology with specific biomarkers. And for this, we really need models that represent the patients. And the lack of well-characterized experimental models is a break on the research in oncology and development of new efficacy anti-cancer drugs. Here I will show you a cartoon that presents the process of the patient-derived xenograft models as a translational research tools. From the clinic, there is clinical needs, and from this we can, with good collaboration with the surgeon, generate in vivo models, directly taking the tumors from the patients and then graft them on mouse models, without any cell culture system in between. And we can amplify such models, and so we can work on we can work on the sorry, it looks like there is a technical issue. We can work on the models and amplify the in vivo pharmacology to test the model. There is a clinical from the models when we test all the compounds on the models, the full collection of models. It's like a phase 1 or phase 2 clinical studies. And then we can go back to the clinical and evaluate if our hypothesis, our preclinical hypothesis, is valid in clinical. From this, we either generate new clinical needs for a new collection, we identify new treatments, new resistance to treatments, and again generate a new in vivo models. The needs for these models need to be very well characterized. That's why we have a full network to collect DNA, RNA, proteins, and serums, and eventually also generate in vitro models from our mouse model collection. All the data are collected in a database, and from that we can perform data mining and identify new biomarker or new correlation with the treatments and the response of the tumor. Our network is really going from in vitro model to the clinical trial, and there is a very close relationship with the surgeon and the oncologist. The focus of OncoDesign and Taconic is really on the products and service. That our brand name is Chemice, which means the collection of patient-derived xenografts. And the way we use it when it is developed with our clients is, first of all, to generate the models for them, and we can provide the full collection and ship to the clients, and there are full rights to use them. Or more in a standard way, we test in a preclinical study the compounds of the clients in our models. Here, the point of doing such new models, the Chemice PDX models, is to have a better evaluation of the targeted therapies compared to immortalized cell lines. We can identify new targets, we can validate them on fresh clinical samples, or on preclinical biological samples, since we are not limited anymore when we work on xenograft models. And such models are very, very well stable. They do represent the histological structure and the molecular characteristics of the tumors. So they are very well set up to validate molecular targets in vivo and to evaluate anti-tumor drug activity in respect to the patient profile. And I want to show you here two analyses, the well-known clinical trial of the KRAS mutation versus KRAS wild type in colorectal cancer. And it has been shown that the KRAS mutation was not leads to a worse prognostic of the patient, whereas with the wild type KRAS, the cetuximab bring a clear benefits in these patients. And this is exactly what we have reproduced with our collection of colorectal models. Here you have the mutated KRAS, and you have here the right type KRAS, and both are treated with cetuximab, and is clear benefits in the subpopulation with the wild type KRAS. So with these two cartoons, of course, this is the preclinical analysis has been done after the clinical analysis, but this is exactly the proof of concept that we hope to do with new compounds that will be tested in our preclinical collection. And so the goal is really to make the proof of concept using a full panel of human tumors. We plan to make better prediction of clinical outcome when we take into account the diversity of each patient tumor phenotype and genotypes, and to identify biomarkers upon response to therapy to stratify the patient for clinical trials. Both can be used then in the clinical assay, and so by doing so, we will reduce the overall cost of clinical development, and our estimation is at least by 80 million. And of course, we will also reduce the timelines, but at least 2 years, by at least 2 years for the full, to reach the full market. And the main goal is to decrease the attrition rate. As you all know, that in cancer, the attrition rate is about 95% of all compounds. So if we can decrease this amount, it's a major issue to make new drugs on the market. Here is a more detailed slide about the process, how we develop the chemotherapy. So we have identification of patients, so it's a direct network with our surgeon that they will collect all the patient clinical history. Of course, a full informed consent of the patient, and there is a serology. We collect the fresh tumor. We have samples from the patients, and then we engraft them on the first 3 mice usually, and we do biobanking at passage 3. The take rate of the tumor is really depending on the subtype of tumors. And usually for colorectal is about 60%, but it can decrease to 5 to 10% for the more difficult tumors. And between P2 and P6, we are running the characterization of the models. Of course, the tumor growth, usually the tumor growth is slower than compared to the standard cell line. And it's a bit faster than in humans, but it's hard to compare. We are performing a full molecular characterization at the transcriptomic or at the chromosomic level. I will show you some data about it, and histological, of course, and pharmacological analysis. Here, this is an example of a pharmacological study with irinotecan or CPT11. So on this slide now, I want to explain you how the collection can really represent the patient diversity. On 86 samples that we collected, this is again colorectal cancer, we collect all from the surgeon, all the tumors that they have seen, and we implanted all of them, and we have 53 models that were successfully engrafted. It's a take rate about 62%, and you can see from the two graphs that they are really match together. And so we have our collection really represents what is seen in clinic. And if we go in more details about the histological and the molecular profile, we have run. Oops, and there is a slide that is missing. Sorry. We have run a characterization on the tumor. You can see on this slide. Here is the tumor of the patient, and here is the xenograft. And after 8 passages, the structure of the tumor is very well conserved. We observe really the same nodules and the same characteristics. And there are several subtypes more or less differentiated in the colorectal tumor, and we do conserve each subtype of the histology that is seen in clinic. Here on the specific labeling, we use an alloprobe which label only the human cells, because one of the main questions that come back very often is, do the stroma is human on mouse? And you can see here, so in the tumor patient, the stroma is blue as well as the tumor cells, whereas in the xenograft models, the tumor cell remain blue, of course, but the stroma is not labeled anymore. So the tumor has been invaded by has been colonized by the mouse cells and fully replacing the human stroma. This occurs usually really at the engraftment at the first passage or second passage. The first molecular analysis is a broad analysis, a CGH array analysis, and on this graph you can see that on blue this is the profile of the patient, and in red this is the tumor graft, and you can see that the profile is very similar. When we have an amplification, this is conserved among the passage, and when we have a deletion, this is also conserved. There is very few difference, and more or less when there are differences, this is in the same order. And to have this conservation to have the genomic stability is very important compared to cell line that usually you don't have such conservation. We have tested also the MSC status when it comes to colorectal cancer, and this is well conserved as well. We can sequence multiple genes. We have run here a study of DNA exon sequencing on the hotspot mutation. So this is the list of genes that we have sequenced. And this is the mutation that we observe on the different exons that have been sequenced. And if we compare to the literature data, so we took the COSMIC database that collects many mutations in the literature, and overall we have a very good match of the mutation between the collection and what is seen in the human clinic. So again, the representativity of the human variability is very good in our colorectal cancer using 50 models. When you go further in the molecular characterization, you can study the gene expression profile, and here we compare the expression between the different samples using an Affymetrix microarray. And on the first graph, we compare the patient tumor versus the first passage engrafted on the mouse models. And most of the genes the expression is very similar, and there are few genes, about 600 genes, that we can observe a slight difference, and all of them are usually correlated with the human stroma, which makes sense because we are losing it. But then over time, when you compare P0 versus P9, the transcription, the profile of the transcription gene is very, very similar, very well conserved. Again, if you compare two tumors engraft, two tumors from the same passage engraft on two different mice, we have a very good conservation of the expression profile. And we have seen that over 15 passages in vivo. Here, so I took this cartoon from a publication in Clinical Cancer Research last year from Capet and all that were comparing the different strategy to evaluate compound before going to clinic. And of course, there is the in vitro cell line, the cell line xenograft, and the patient-derived xenograft, which is a real link between the standard models and the patient in clinic. And so, of course, the heterogeneity is one major point that you will see in our collection that you don't have with cell line. You have a very good conservation of the molecular subtype, which is very important, especially when you want to study targeted therapies. You can have here it is highlight a mixed stroma and murine and human. It is known that in some models, some tumor, the human stroma might be kept a little bit longer, but it's still a debate about this. In our collection, so far, we have mostly seen murine stroma. Nevertheless, we can envision to engraft as well some human cells, either for some fibroblast human cells, some CAPS cells, or even the immune system. This is also something that is doable. We observe a slower growth. It is not as slow as that is seen in humans, but this is much representative than the rapid growth of the double cell line. There is the main difference also is that our tumors. The main difference is that tumors usually sometimes have been treated with patients before, and which is not the case with cell lines. So we can address some resistance that have been seen in some patients, and we can address this in our collection. And I won't go any further, but just to make short, we have primary and metastatic sites. We can do some orthotopic implantation and so on. Just now, I want to go to chemotherapy. So in our collection of colorectal cancer, we have tested the four main treatment that is used in clinic: the 5-FU, the oxaliplatin, the irinotecan, and the cetuximab. So 3 drugs and an antibody. This is the scheme of treatment that we have used, and as a monotherapy each time. The mice have been randomized, of course, between 100 and 200 cubic millimeter, for the rats between 300 and 400, because we can do both skin mice or nude rats. This table recapitulates all the models that we have collected, all the 86 models, the 86 patients, and the including 53 mouse that have been engrafted. If we zoom on it, on this table, so you have all the name of the tumor. When there is a box like here, it means it's coming from the same patient. So we have a primary tumor and a metastatic tumor. We have the mutation in pink and the wild type gene. So this is the mutation. This is the MSI status, if it's low or high, if these models have been successfully engrafted or not. If we have performed a CGH analysis, some other mutation have been done, and just as I mentioned, the therapeutic responses have been seen in these models. So for 5-FU, oxaliplatin, irinotecan, and cetuximab. So the 4 drugs have been tested individually on all the models. We're using a group of 10 mice each time that have been randomized. And so when you have a minus, it means that the drug has no effect at all. This means a TCOC that is above 42%, which is the standard used at the FCI to classify the drug. With a plus, it means that the standard response has been between 10 and 42% of the TCOC. 2 plus is below 10%, and 3 plus, it means also that we have obviously below 10%, and we have also seen a regression of the tumor. And if you look at all, you can see that with 50 models, you have a huge difference in therapeutic response. And this is what you have in clinic. So when we do a preclinical study on one cell lines, of course, it does not match what we will observe in clinic. And doing a full study on numerous models really help us to classify and stratify the patients that you might select for your clinical trial later on. Here is I will go to more details about the study that we ran for Cetuximab sensitivity versus KRAS status mutation. So as you might probably know, KRAS is a downstream EGFR. It is part of this pathway, and when you have this pathway mutated, most of the time is KRAS mutation. The patient will be resistant to Cetuximab, and this is what we have in these models. The 22, you have a mutation of KRAS, and the Cetuximab is not efficient at all. On the other hand, when the KRAS is wild type, the Cetuximab will give us a very good response with the full regression of the tumor. Nevertheless, we have some KRAS mutating models that have been identified, actually 4 out of 19. And this means that there are some patients that now are not treated with Cetuximab because they do not harbor a KRAS mutation that might benefit of the Cetuximab. And our models can be a good way to understand which population is KRAS mutated and sensitive to Cetuximab. This is one of the examples that we can run with such models. And as I introduced you early on, this is more or less the same study. Here, this is the mice that have been treated with Cetuximab or with control with KRAS mutation, and there is no clear benefit. When you do the same study taking into account only the wild type KRAS models, there is a clear benefit of the Cetuximab response. So again, we can run. Here we took all the 53 models that we have done. We tested all the Cetuximab in all models, and clearly we reproduced the data that has been shown in clinic. So we really think that we have a high value of this model for the design of clinical phase 2 studies. So we focus on the KRAS mutation, but in the EGFR pathway there is of course other genes. So in our models, if you took all the EGFR mutation that we have seen, and of course we have not looked probably not at all mutation possible, but we have 17 models that do not respond, which is expected, but there are 4 models with mutations, among them 3 are KRAS mutated here and here, that do respond to Cetuximab, and so this is very striking in terms of clinical response, and maybe some population could benefit from a further study of these models. On the other hand, we have models that have a full EGFR pathway, wild-type pathway, at least for the gene that we have looked for, and 10 do not respond to Cetuximab. So again, with these models, we can look for the population that do not respond to Cetuximab, are resistant to Cetuximab, and eventually either identify new drugs or make new biomarker that will have a high value in clinic. Such study has also been conducted in other groups. As an example, everolimus has been put in clinical study after mouse model studies that has been published by Raymond and Yao. And now there is a phase 3 clinic on the combination of sunitinib and everolimus based on the results that have been done in preclinical studies. This is for pancreatic neuroendocrine tumor, as you all know, this is very deadly cancer. So the preclinical trials on full cohort of mice that are engineered to develop a particular type of drug is a good starting point to evaluate the mechanism-based drug. And it helps to guide the design of the clinical trial for the same type of cancer, and there is a clear clinical benefit that can be seen in preclinical, such as progression-free survival or overall survival that is needed then to justify the drug approval for clinical use. And the relapse and clinical failure can be, again, then study in preclinical trials. This is the first cartoon that we showed. When we identify new clinical needs, we can generate new models and understand the limitation of the first trial. All the work that has been presented so far on the colorectal cancer has been published in more detail. This was part of the Genomic consortium that we've been part of, that we actually were the leader of the group. And this publication, this article has been published in Clinical Cancer Research last October. So if you want full details, we'll be happy to share the data, but you can also find them in this publication. I will just go very quickly through the next slide. This is an example of the data that we'll share when you purchase a model at Taconic or CoDesign. So this is an example of one of the model that we call CRIC-004M. This is a metastatic model. So this is the patient clinical history. So it's important to make some correlation with the tumor. So it was an hepatic metastasis. The patient also had prostate cancer and a colon cancer. There is a histological nature. We know that it's well differentiated, and there is a treatment that has been done on this patient, and the outcome when it's available. Then we have the first passage. So here it's P4 on skin mice. Here it's P6, and you have an example of the growth rate of the tumor here. This tumor is quite fast. So we can share the table with the take rate and the doubling time, and the time to reach the randomization, and the time to reach the endpoint. So here is the slide that I wanted to show you initially that didn't work. Was the tumor histological structure. So here it's with the patients, the skin mice, and you can see that the structure is very well preserved. And we have a full analysis of the histological structure on this table. I won't go into the detail. It's just to show you, to highlight that it's very well conserved. The well differentiation profile of this tumor is conserved over time. In mice, we can usually observe a decrease in the percent of stroma, but it remains quite high. In rats, the stroma is closer, the amount of stroma is closer than in humans. The genetic mutation of the MSC status of this specific tumor, and we have done usually some biomarker analysis. Here the CEAs, the EGFR expression, and some hypoxia gene, some blood vessel biomarker is also sometimes that we are doing. The CGH array with the deletion, the gain or loss of copy, and the genes that are usually known to be in the region that were lost or gained. We can perform some ex vivo analysis that's been done on this specific tumor, and the results for the pharmacological study that's been done in vivo. So here as an example, it was not responsive to oxaliplatin, 5-FU, and we get a response with CPT11 and a very good response with Cetuximab. So here is the graph of this study, and so we have tested, as you can see. So in blue, it is animals that have been treated. In black, the animals are control animals, and so there are two drugs that are inactive, and the Cetuximab is very active. Here on this slide, we show the data to compare the mouse and the rats. So here you have the CPT11, and we have the other drugs on the skin mice. And if you go to cytotoxic like CPT11, you can see that the response is the same, or for the 5-FU oxaliplatin, we have no response as it has been seen. Nevertheless, we observe a difference between mouse and rats for the antibody here, most likely because the tumor can grow longer, and there is probably the treatment needs more time to be efficient, and by doing so, we can see that cetuximab is more efficient in these models that it is in the other in the mice. Here, this is the collection of the colorectal collection. This is mainly what I focus on today. I should tell you that we are working on many other subtypes of cancer, myeloma, AML, liver, pancreas, and so on. And their collections are ongoing. Notably, the breast collection is well advanced now. And we have on the blue is the one that are already available for study at a co-design or for sale. Just to summarize what I have tell you today, so we have the network to collect all the tumor directly from the surgeon, to associate them with the clinical data. We generate the PDX models on the appropriate strain, usually the skin on the nude mice. We can go further down with immunodeficient mice, the NOG1. This is notably used if you want to engraft other cell types like immune cells or stroma cells. We will characterize these models or send to you directly if you wish to get only the models and test then the compounds. So there is a PDX shipment that is available as a service offer, or the preclinical study that can be performed here at co-design. We can validate the targets in our models depending on the transcription gene that are available, and look for the model that fits your targets. We can run the preclinical study and do the drug evaluation or biomarker identification. And a good thing we try to work on low passage sample that will retain the heterogeneity and the fidelity of human cancer. All the studies that I have shown you today were done on tumor that were below 10 passage. We don't know, the structure, histological structure remain good for a longer time, but there is not much study that has been done on human tumor that, over 40 or 50 passage. This is something that is not done yet. We have data that full data that are available to guide the selection of model. Of course, most of the time you don't want to start right away with 50 models, so you will work with 5 or with 3 to 5 models, and if it works, you can then set up a full study to guide your clinical trial. And there is many, as I mentioned, many new PDX collection in development for multiple cancer pathology. Most of you have your own specific pathology, your own specific request. This is also why we are usually working with you to develop a collection that matches your needs, rather than always providing tumor that has models that are fully ready. Okay, this is all for today, and I hope I've been clear to explain you the benefits of all these PDX models, and if there are any questions, I will answer to them.
Okay, Cyril, thank you very much for this detailed presentation and the examples of how to use PDX models in drug testing and how to align them to clinical trials. Indeed, we do have actually a rather large number of questions, so I would propose, Cyril, that we just go into the questions that have come in here over the chat room, and I will address them in the order that they came in. As mentioned before, if we're not providing the answer today to a question, we will follow up via email with you. So the first question, Cyril, that came in was the one here. Is there a difference in tumor growth in ectopic versus is there a difference in tumor growth in ectopic versus orthotopic site grafting?
Yeah, we don't have extensive experience with orthotopic models for PDX orthotopic models. What we have observed is that we do get a growth, and at the beginning it looks more or less similar, but we reach a plateau at one point. So at least for colorectal cancer, at 400 or 500 cubic millimeter, it don't go beyond this volume. So early on it's fine, but later on, then it depends on each pathology, and I might not have a clear answer.
Do you have experience with other tumor types that you compared ectopic versus orthotopic grafting in breast cancer, for example?
For breast cancer, it's more or less the same thing, because when you engraft in subcutaneously or in mammary fat pad, there is not major difference. So for breast cancer, it should be fine. And what we try to work on is to work on with metastasis. So when we engraft primary tumor, we have seen some metastatic tumor. We have about 12 models with a colorectal collection that we were able to observe a metastasis from the primary tumor.
Okay, good. Let's move on to the next question. That is, what is the difference in tumor doubling time in mice compared to that in humans?
As I say, and it was shown on one of the slides, usually in humans, the doubling time is about in months, where here we will be in weeks, which is slower than standard cell line, but faster than in humans. It's between.
Okay. The next question is a long one and rather complex, I believe, but I'm just reading it, and then we can address part by part. What about the immunological response? What mice or what mouse backgrounds do you have? Do you have models to assess the immunological response too?
So most of the models are done in SCID or immune mice, which are immunodeficient animal models, but we provide more sophisticated models, meaning that we can work also on NOG mouse models, and then engraft the immune system, so we can combine the PDX models with a full humanized mouse models, and then recapitulate the immune response.
Right. Yeah. Okay. This question continues, and that is referring to the stroma now. So the same for stroma. You are assessing mouse stroma response interactions and not human. Is that considered as a problem?
We don't think it is a problem. Of course, we might have a difference. We cannot. This is mouse stroma. It's not human stroma. But in terms of vessels, in terms of nutrients and that is bring to the tumor, I think we can consider that it's quite close. And if you look at the histology, the structure remains the same. As I said, in mice we have a lower amount of stroma. In rats, it's closer than in humans. So there are differences. We do not understand completely yet.
Okay, I'm sure this is a complex question that can be discussed further, but thanks for the initial response. The next question that we received is the following. Is there a difference in tumor... No, we had this already, I'm sorry. Yeah, here, this one. Do you see mutations appearing upon treatment? Are you assessing this?
This is a very good question, but we never assess in our models the occurrence of new tumor, of new mutation after treatment. This is something that could be done, develop new resistance in our models, but we usually prefer to work directly from the resistance that has been seen in patients. This is more the way we are working. But this could be done, I guess.
Mm-hmm. Right, but it has not been done yet, right, at this point?
No.
Yeah. Okay, there is a question in a similar area. We have, just to let you know, we have 3 more questions now, so this is the third to last question. Can you see a correlation between previous patient treatments and the response to chemotherapy or antibody treatment in your models? So basically, I understand how well is the patient response mimicked in the mouse model, PDX mouse model?
Usually we have a quite good correlation. It's hard to give a clear answer because most of the patients do not have a monotherapies, and we test our model so far in monotherapy, so it's not always easy to make a clear link. But for the ones, Cetuximab is probably the easiest to do. There we have usually a good correlation. 5-FU and oxaliplatin are usually, you give FOLFIRI, you give you give the 3 therapies together, so it's not always easy to compare the clinical outcome with the model.
Okay. The next question is also interesting, and this is also, I think, a very practical question. First, the question starts as follows. There is large variability between models, which is very interesting, and again, I think this is reflecting the individual patients. The question is the following then. How about variability within a given model or tumor line? How homogeneous are the different animals engrafted with a given sample? Linked to that, how many mice engrafted with a given tumor do you need or condition for a specific readout?
So there is an heterogeneity of the models. We did not assess specific subcompartment of each tumor. Nevertheless, when you look at the transcription profile, this is quite homogeneous between the different passages and between tumors engrafted on different mice. So this heterogeneity that we observe is conserved over time. Usually we work with such study with 7 to 10 mice in preclinical study, and this is good to get a good answer in terms of statistics.
Okay, thank you. And then the final question that we have for today is: can you achieve serial passaging with AML, acute myeloid leukemia, PDX, and do you have non-invasive biomarkers to track AML engraftment?
This is a program that is ongoing. I know that we have been successful in our Genomic partnership to generate such models. I'm not truly the expert in this pathology, so I will not get into the details. But there is an ongoing program at Oncodesign for AML PDX. And if there is a specific question, we can address that to the expert in the lab here.
Okay. Okay, then I think we have spent 45 minutes now learning about PDX models. Cyril, I want to thank you again and your team in France for not for hosting, but for participating and offering today's webinar. And I want to thank all of the people that dial in today and listen to us, and especially our colleagues in Japan. I saw there were a number of participants from Japan. Thank you for staying up so late and joining us. And thank you very much. We will be following up with another seminar probably in about 2 months' time, and we'll inform you appropriately. Have a good day. Thank you.
Thank you.